Good morning and a very warm welcome to all. So in the previous class we have discussed about different classifications of the bacteria based on their size, shape and arrangement. Now we can discuss each morphological details of the bacteria. Okay. So actually this bacteria, it is a single cell organism. And the cell structure of the bacteria, it is similar than that of other organisms, and it doesn't possess nucleus or membrane bound organelles. Instead, their control center contain genetic information, and it is contained in a single loop of DNA. So it doesn't possess any nucleus. Some bacteria have an extra circle of genetic material, and it is called plasmid. The plasmid often contains genes that give bacteria some advantage, such as antibiotic resistant mechanism, etc., over other bacteria. For example, it may contain a gene that makes the bacterium resistant to antibiotic. And we have already studied that bacteria was classified into 5 groups according to their basic shapes. That is spherical, rod-shaped, spiral-shaped, comma-shaped, then corkscrew, that is spirochetes. They can exist as either in single cells or in pairs or chains or clusters. And also these bacteria are found in every habitat on earth, such as soil, rock, oceans and even the Arctic snow. Some live in or on other organisms, including plants. Some bacteria can be seen along with plants and animals, including humans. There are approximately 10 times as many bacterial cells as the human cells in the human body. A lot of these bacterial cells, these are also found lining the digestive system. Some bacteria can be seen in the soil or on the dead plant matter, and they play important role in the cycling of nutrients. Some bacteria are responsible for causing food spoilage and crop damage, and some of them are incredibly useful in the production of fermented foods such as curd, yogurt and soy sauce. Relatively, a few bacteria are pathogens or parasites that cause disease in animals and plants. Now, this is the picture of comparison of a bacteria. I think the bacterial structure is not much visible, but this picture is of the eukaryotic cell, that is a plant cell and an animal cell. In this both plant cells and animal cells possess different types of membrane bound organelles such as nucleus, chromosomes, mitochondria, endoplasmic reticulum, chloroplast, Golgi bodies, etc. But these structures are absent in bacterial cell. Now coming to the we can go through detail about the each morphological details of the bacteria. First of all, we can discuss about the bacterial cell wall. The bacteria are protected by a rigid cell wall, and it is composed of peptidoglycans. So the major component of this bacterial cell wall, it is peptidoglycan. Actually, this bacteria lack membrane bound nucleus, and their genetic material is also naked within the cytoplasm, and ribosomes are their only type of organelle. So any other types of organelles are absent, and this nucleoid portion, it is seen in the bacteria, and it refers to the region of cytoplasm where the chromosomal DNA is located, usually either in single or circular in chromosome. So the cell wall of the bacteria provide protection to the bacteria. And also these are certain features that bacteria reproduce by means of binary fission, duplicating their genetic material and essentially splitting to form daughter cells identical to the parent. And about the bacterial cell wall, it is located outside the cell membrane. So it can be seen outside the cell membrane, and it may provide the bacteria cell support and protection against mechanical stress or damage from osmotic rupture and lysis. So it protects the bacteria from this sort of osmotic rupture and lysis. The major component of this bacterial cell wall is peptidoglycan or murine. This rigid structure of peptidoglycan specific only to prokaryotes. So this feature peptidoglycan, it is specific only to prokaryotes. That means in the bacteria. It gives the cell shape and surrounds the cytoplasmic membrane. So it can be seen around the cytoplasmic membrane. And what do you mean by peptidoglycan? It is a huge polymer of disaccharides. That means glycan. And they are cross-linked by short chain of identical amino acid monomers. So it is a huge polymer of disaccharides, and those disaccharides are cross-linked by short chain of identical amino acids monomers. And the backbone of this peptidoglycan, the name itself, peptido means amino acid, that is protein, peptide. Glycan means disaccharide. So both, combining both. So the backbone of the peptidoglycan molecule, it composed of two derivatives of glucose. So what are the two derivatives of glucose present in the peptidoglycan? NAM and NAG, short form. NAG means N-acetyl glucosamine. NAM means N-acetyl muramic acid. And these are cross-linked with a pentapeptide coming of NAM and varying slightly among the bacteria. The NAM and NAG strands are synthesized in the cytosol of the bacteria. So they are synthesized in the cytosol. Now, this NAM and NAG, they are connected by interpeptide bridges. Okay, and they are transported across the cytoplasmic membrane. So synthesis takes place in the cytosol. After the synthesis, they are transported to the cytoplasmic membrane by a carrier molecule. The name of that carrier molecule is bactoprenol. Okay. Now they are present in the above the cytoplasmic membrane. From the peptidoglycan inwards, all the bacterial cells are very similar. Going further out, the bacterial world divides into two major classes. So, apart from the peptidoglycan, most of the bacteria appear in similar. So, important, this step is important, that is based on the peptidoglycan structure, which is either thick or thin. Based on this, we can divide the bacterial world into two major classes. That means gram positive and gram negative. So this gram positive and gram negative is mainly based on the thickness of the peptidoglycan. Now the cell wall provides important ligands. For what? What are the other functions? For the adherence and receptor sites for viruses or antibiotics. So these antibiotics usually bind onto the bacteria by binding onto certain ligands which are present within the cell wall. Also this virus also bind with certain ligands present within the bacteria, that is present within the cell wall of the bacteria. Now coming to the gram negative outer membrane. So two different types of bacteria present based on their thickness of cell wall. First is gram negative. So this gram negative and gram positive can be differentiated according to the staining procedure. That means gram positive bacteria, it appears purple in color because of the more thickened cell wall, that we will study later. Then gram negative bacteria has thinner peptidoglycan layer. So we can study about gram negative outer membrane. The gram-negative cell wall, it is mainly composed of three important components. That means an outer membrane, a peptidoglycan layer, and a periplasm. So these are the three important components which are present within the gram-negative cell wall. Outer membrane, which can be seen, outer portion, then a peptidoglycan layer, and also there is an inner membrane, and also there is a periplasm. Okay. In the gram-negative bacteria, the cell wall is composed of single layer of peptidoglycan. So in gram-negative bacteria, the peptidoglycan layer is single layer and it is surrounded by a membrane structure, and that membrane structure is called what? Outer membrane. Okay. The gram-negative bacteria, they can't retain the crystal violet stain in the gram staining procedure, but they are able to retain the counter stain, that adding last. Okay. That is commonly the counter stain used is safranin, which is added after the crystal violet. That means after the washing with alcohol, acid alcohol wash, then after last we are adding counter stain safranin. So the safranin, it is responsible for the red or pink color which are seen in the gram-negative bacteria. So gram-negative bacteria appear as pink in color because of the counter stain, that is either safranin or any other. Then gram-negative cell wall, it is very thinner, that is 10 nanometers thick and less compact than that of the gram-positive. That is important point, but remains strong, tough and elastic to give them shape to the bacteria and protect them against extreme environmental conditions. Even to the outer membrane of this gram-negative bacteria. So in the case of the gram-negative bacteria, most important pathogenic unique component is what? Outer membrane. And this outer membrane contain lipopolysaccharides in addition to proteins and what? Phospholipids. So this outer membrane contain lipopolysaccharides. Okay. This lipopolysaccharides, apart from the proteins and phospholipids, that is the most important component of the gram-negative bacteria. And this lipopolysaccharide molecule, it is very toxic and it is classified as an endotoxin. So this bacteria produces two types of toxin, that is exotoxin and endotoxins. These endotoxins are mainly produced by the gram-negative bacteria, that is within the outer membrane. This endotoxin, that is lipopolysaccharides, are located. So they are capable of eliciting a strong immune response when the bacteria infect animals. So this endotoxin is responsible for eliciting what? A strong immune response. So it is very toxic. In gram-negative outer membrane, that means in the gram-negative bacteria, it is usually thought of as a part of outer leaflet of the membrane structure, and it is relatively permeable. So this outer membrane is relatively permeable. It contains structures that helps the bacteria to adhere to animal cells and cause disease. So this outer membrane, it may possess certain structures that helps the bacteria to attach to the animal cells and to cause disease. And the peptidoglycan layer, it is non-covalently, they are anchored to the lipoprotein molecule called bronze lipoprotein through their hydrophobic head. So these are non-covalently anchored to the outer membrane, what? Peptidoglycan layer, and it is through the bronze lipoprotein. Okay. Sandwiched between the outer membrane and plasma membrane, a concentrated gel-like matrix, it is found within the periplasmic space. Look the picture. This is called periplasm. So in between this outer membrane and this peptidoglycan layer, there is a periplasmic space which is filled with a fluid called periplasm. It is in fact an integral component of the gram-negative cell wall, and it contains binding proteins for amino acids then sugars,